Cauliflower mosaic virus, CaMV, is a member of the genus Caulimovirus, one of the 6 genera in the family Caulimoviridae, which are pararetroviruses that infect plants. Pararetroviruses replicate through reverse transcription just like retroviruses, but the viral particles contain DNA instead of RNA. Definition: aphid species Mysis persicae. The cauliflower mosaic virus, CaMV, is a member of the family Caulimoviridae. This family is grouped together with the Belpowviridae, Metaviridae, Pseudoviridae, and Retroviridae, all of which instead have an RNA genome replicated via a DNA intermediate. In the order Virales, the Hepidnaviridae, despite having a DNA genome replicated via an RNA intermediate, like the Caulimoviridae, are more distantly related, belonging to the separate order Virales. Both orders belong to the same class, the Vector-Viruses. CaMV infects mostly plants of the family Brassicaceae, such as cauliflower and turnip, but some CaMV strains, D4 and W260, are also able to infect Solanaceae species of the genera Datura and Nicotiana. CaMV induces a variety of systemic symptoms such as mosaic, necrotic lesions on leaf surfaces, stunted growth, and deformation of the overall plant structure. The symptoms exhibited vary depending on the viral strain, host ecotype, and environmental conditions. CaMV is transmitted in a non-circulatory manner by aphid species such as Mysis persicae. Once introduced within a plant host cell, virions migrate to the nuclear envelope of the plant cell. Structure: the CaMV particle is an icosahedron with a diameter of 52 nanometers built from 420 capsid protein, CP, subunits arranged with a triangulation T = 7, which surrounds a solvent-filled central cavity. CaMV contains a circular double-stranded DNA molecule of about 8.0 kilobases, interrupted by nicks that result from the actions of RNase H during reverse transcription. These nicks come from the MET tRNA, and 2 RNA primers used in reverse transcription. After entering the host cell, these single-stranded and quat nicks and quat in the viral DNA are repaired, forming a supercoiled molecule that binds to histones. This DNA is transcribed into a full-length, terminally redundant 35 sRNA and a subgenomic 19 sRNA genome. The promoter of the 35 sRNA is a very strong constitutive promoter responsible for the transcription of the whole CaMV genome. It is well known for its use in plant transformation. It causes high levels of gene expression in dicot plants. However, it is less effective in monocots, especially in cereals. The differences in behavior are probably due to differences in quality and/or quantity of regulatory factors. Recent study has indicated that the CaMV35S promoter is also functional in some animal cells, although the promoter elements used are different from those in plants. While this promoter had low activity compared to canonical animal promoters, levels of reporter products were significant. This observation suggests that the 35S promoter may have potential for use in animals. The promoter was named CaMV35S promoter, and quat 35S promoter and quat winking face, because the coefficient of sedimentation of the viral transcript, whose expression is naturally driven by this promoter, is 35S. It is one of the most widely used, general-purpose constitutive promoters. It was discovered at the beginning of the 1980s by Chua and collaborators at the Rockefeller University. The 35S RNA is particularly complex, containing a highly structured 600 nucleotide long leader sequence with 6 to 8 short Open Reading Frames, ORFs. Genomic map of CaMV. This leader is followed by 7 tightly arranged, longer ORFs that encode all the viral proteins. The mechanism of expression of these proteins is unique, in that the ORF-V protein, encoded by the 19S RNA, controls translation. Reinitiation of major open reading frames on the polycistronic 35S RNA, a process that normally only happens on bacterial mRNAs. TAV function depends on its association with polysomes in eukaryotic initiation factor EIF3, ORF-I, P1, movement protein, PO3545, ORF2, P2. Aphid, insect transmission factor, PO3548, ORF3, P3. Virion-associated protein, VAP, PO3551. Structural protein, DNA binding capabilities ORF-IV, P4. Capsid protein, CP, PO3542, ORF-V. P5. Pro-pol, PO3554. Protease, bifunctional reverse transcriptase and RNase H ORF-V, P6. Transactivator, viroplasmin, PO3559, inclusion body formation, trafficking, possibly other functions. See text, ORF7, V. Unknown. Appears note to be required for infection, Q83163. Q83164, contains a tRNA MET binding site in addition to its functions regarding translational activation and formation of inclusion bodies. P6 has been shown to interact with a number of other CaMV proteins, such as P2 and P3, suggesting that it may also contribute in some degree to viral assembly and aphid-mediated transmission. In addition, P6 has been shown to bind to P7. Investigating interactions between the two may help to elucidate the as yet unknown function of P7. Another function of P6 involves modification of host non-expressor of pathogenesis-related 1,1-Nepalese rupee during the course of infection. NPR1 is an important regulator of salicylic acid, SA, and jasmonic acid, YA, dependent signaling, and is most closely associated with crosstalk between the two. Modification of 1-Nepalese rupee serves to inhibit plant cells' defensive responses by preventing SA-dependent signaling. Modified 1-Nepalese rupee can properly traffic to the nucleus and bind the PR1 promoter, but is unable to initiate transcription. Because active 1-Nepalese rupee is required for accumulation of SA, this leads to a further depletion of SA. Whereas regulation of SA-dependent signaling by P6 modified 1-Nepalese rupee is localized to the nucleus, regulation of YA-dependent signaling is cytoplasmic in nature and involves the COI1 pathway. In contrast to that of SA, YA-dependent signaling is increased in the presence of modified 1-Nepalese rupee. Replication. A diagram depicting the steps in the genome replication of cauliflower mosaic virus, CaMV. DNA is depicted in blue and RNA, including the tRNA, is depicted in red. See text for more details. CaMV replicates by reverse transcription. Viral particles enter a plant cell and are unencapsulated. At this stage the viral DNA consists of 3 fragments, 1 on the strand, alpha, and 2 on the strand, beta and gamma, which are imperfectly assembled into a circular genome with 3 gaps or discontinuities, D1, D2, and D3. The viral DNA enters the nucleus where the discontinuities are filled in. At this point, the viral DNA also associates with host histones, forming a mini chromosome, not shown. The host DNA-dependent RNA polymerase transcribes from the 35S promoter all the way around the viral genome, surpassing the 35S promoter. This creates 2 copies of the 35S promoter in the resulting RNA. Transcription also initiates at the 19S promoter, not shown. The viral RNAs pass into the host cytoplasm where they are transcribed. The 3' end of a tRNF met anneals to a site corresponding to discontinuity 1, D1, near the 5' end of the 35S RNA. The tRNF met primes synthesis by the viral reverse transcriptase, encoded by ORF-V, of a new alpha strand. RNase-H removes the RNA from the DNA-RNA duplex, leaving behind the DNA. This new DNA binds the 35S promoter at the 3' end of the RNA template, and synthesis of the alpha strand of DNA continues, and RNase-H continues to degrade RNA complex to DNA. Synthesis of the alpha strand completes. RNase-H activity exposes purine-rich regions at the position of discontinuity 3, D3, which primes the synthesis of the gamma DNA strand. RNase-H activity exposes purine-rich regions at the position of discontinuity 2, D2, which primes the synthesis of the beta DNA strand. When the new gamma strand of DNA reaches the 5' end of the new alpha strand, it switches to the 5' end of the new alpha strand, recreating discontinuity 1, D1. When the new gamma strand of DNA reaches the 5' end of the new beta strand, it displaces the primer and some of the newly synthesized beta strand, resulting in the recreation of discontinuity 2, D2. When the new beta strand of DNA reaches the 5' end of the new gamma strand, it displaces the primer and some of the newly synthesized gamma strand, resulting in the recreation of discontinuity 3, D3. At this point, the new viral genome can either be packaged into capsids and released from the cell, or they can be transported by movement proteins into an adjacent, uninfected cell. 14. The Cauliflower Mosaic virus promoter, CaMV35S, is used in most transgenic crops to activate foreign genes which have been artificially inserted into the host plant. It is inserted into transgenic plants in a form which is different from that found when it is present in its natural brassica plant hosts. This enables it to operate in a wide range of host organism environments which would otherwise not be possible. CaMV contains about 8 kilobits double-strand DNA genome and produces spherical particles. CaMV infections are systemic, and even its DNA is infectious when inoculated on abraded plant surfaces. The CaMV genome has 8 tightly packed genes, of which only 2 small genes 2 and 7, are non-essential. As a result, only these 2 genes can be replaced, deleted without a loss of infectivity. In addition, modified CaMV genomes exceeding the natural genome size, 8024 BP, by even a few hundred BP are not packaged into virions. These 2 factors seriously limit the size of DNA insert clonable in CaMV. The bacterial dihydrofolate reductase DHFR gene has been successfully cloned into the CaMV genome, in place of gene 2, and has been successfully expressed in plants. Molecular mechanisms of vector-mediated CaMV transmission. The virus is acquired from an infected host during feeding by the aphid vector. 2. A transmissible complex is composed of virions and protein P2 located in the vector's stylets. The P2N terminal domain recognizes a protein receptor located at the tip of the stylet, and the P2C terminal domain binds to the P3 decorated virions. Transmissible complex of CaMV. The mode of acquisition by the vector is controlled by the tissue and intracellular specific localization of P2. This protein is only found in epidermis and parenchyma cells. Moreover, in these cells, P2 is localized in single viral electron-lucent inclusion bodies, ELIB, in host cells. Viral protein P2 and P3 are first produced in numerous viral factories, electron-dense inclusion bodies, and are later exported and co-localize with microtubules, before concentrating in ELIB. CaMV specifically uses the microtubules to form the transmissible body and thus enable vector transmission. The complete molecular characterization and study of this virus was not carried. Further, evasion of plant defenses. Cauliflower mosaic virus possesses a number of mechanisms that allow it to counteract host plant cell defenses. While the pregenomic 35S RNA is responsible for genome replication by reverse transcriptase, it also contains a non-coding 600 base pair leader sequence that serves as an important mRNA for the production of factors involved in viral counter-defense. A number of hosts of CaMV possess small RNA-based viral silencing mechanisms that serve to limit viral infection. The products of the aforementioned 600 BP sequence are viral small RNAs, vsRNA, of 21, 22, and 24 nucleotides in length that serve as decoys, binding and inactivating effectors of host silencing machinery, such as Argonaute-1, AGO1. As proof of principle, experimental overexpression of these vsRNAs allows for increased viral accumulation in infected plants. Concerns about use of CaMV35S promoter in transgenic plants. In the early 2010s, some concerns have been raised about using the CaMV35S promoter for expression in transgenic plants. Because sequence overlap exists between this promoter and the coding sequences of P6. 54. Transgenic events certified for release in the USA contain up to 528 BP of ORF-V, encoding C-terminal domains of P6. As P6 is a multifunctional protein whose full range of functions is unknown, there is some concern that expression of one or more of its domains may have unforeseen consequences in the transgenic organisms. Recent studies have attempted to determine what length of CaMV35S promoter has the least chance of inadvertently producing P6 domains, while still retaining full promoter activity. As one might expect, using shorter promoter lengths decreases the number of P6 domains included and also decreases the likelihood of unwanted effects.